So at first, we will discuss about what is carbohydrate and what are the types of carbohydrate. So carbohydrates are basically polyhydroxyaldehyde ketones compounds which are the most abundant biomolecules present on earth. So what are the roles of aldehyde and ketones in the procedure in the creation of carbohydrates? That we'll discuss later in this video. So now if we take a look at the general formula of carbohydrate, we can get that is CnH2O whole N. So here N represents a particular number that can be 1, 2, 3, 4, 5, 6, any other numbers. So now if we take an example for understanding the formula of carbohydrate, we take a example that is glucose, which has the formula of C6H12O6. So this formula can be written in this manner also, that is the C6H2O whole bracket 6. Okay, so here N is equals to 6. So if we break the bracket, then we can get the previous formula, that is the C6H12O6. So thus we can create any formula of any carbohydrates by using this general formula, that is the CnH2O whole N. So now let's discuss the types of carbohydrates. So carbohydrates has mainly three types, that are monosaccharide, disaccharide and polysaccharide. Monosaccharide is the carbohydrates that have the only one single sugar molecule. Mono means single and saccharide means sugar molecule. So here in the monosaccharide, the carbohydrate has a single sugar molecule. As for example, we can take the glucose, fructose, galactose, and for the disaccharide, di means two and saccharide means sugar molecules. So here two sugar molecules linked with each other by a specific bond that we'll discuss later in this video. As for example, we can take maltose, sucrose, lactose, etc. And for polysaccharide means many sugar molecules, that means more than two sugar molecules linked with each other. And as for example, we can take starch, glycogen, cellulose, etc. Okay. So now let's discuss briefly the all three types of carbohydrates, that are monosaccharides, disaccharides and polysaccharides. So at first, monosaccharides. Monosaccharides, we previously discussed that they have the single sugar molecule, and the most abundant monosaccharide is glucose. Okay, so we can see that now that monosaccharides have multiple number of carbons, that are 3 carbon, 4 carbon, 5 carbon, 6 carbon. You can see in the pictures, and some examples of the monosaccharides are such as 3 carbon monosaccharides is the glyceraldehyde, and then 4 carbon is the erythrose, and 5 carbon is the ribose, and the 6 carbon is the glucose. So now here the monosaccharides are differentiated in two types, that are called aldose and ketose. You can see that the previous four examples, you can see the functional group is aldehyde. Okay, and the last one has the functional group as a carbonyl group or a CO group. So those monosaccharides have the aldehyde group or the CHO group as their functional group. Those monosaccharides are called as aldose. Okay, and those monosaccharides which has the carbonyl or CO group as their functional group, those are called ketose. Okay, so in this example we can see that dihydroxyacetone, which has the 3 carbon structure and has the carbonyl or CO group as their functional group, and so it is called as the ketose monosaccharide. Okay, so thus we can differentiate between aldose and ketose monosaccharide because of their functional group. Okay, now let's talk about the disaccharides. So as we previously said that the disaccharides is formed when the two sugar molecules are binded with each other by a specific glycosidic bond. Okay, so what is the glycosidic bond? It is a bond formed between the anomeric carbon atom of a monosaccharide and the oxygen atom of an alcohol. Okay, so in this example we can see that glucose plus fructose is equals to sucrose. So here glucose and fructose are two monosaccharides and they bind with each other by the help of the glycosidic bond, which you can see that the sucrose structure. Okay, the intermediate bond between the glucose and fructose structure. And if we want to know that what is the most abundant disaccharide, then the answer is sucrose. Okay, so how the sucrose is formed, this is discussed, and now come into the polysaccharide. Now for polysaccharide, we all know that polysaccharide is formed when many sugar molecules are binded with each other, and then the polysaccharides are formed. So for the disaccharide, only two monosaccharides are binded with each other, and for the polysaccharides, many. That means more than two sugar molecules are binded with each other and form the polysaccharide. So this is a very ubiquitous in nature. That means it founded in everywhere. Now, these polysaccharides have the two major types that are called as the homopolysaccharide and the heteropolysaccharide. So the homopolysaccharide are the polysaccharide that have the only single type of monomeric unit. In the picture, you can see that the all yellow units. That means if the unit is glucose, then the all units will be the glucose. Okay. So these homopolysaccharides are structurally of two types that are called branched and unbranched, as you can see in the picture. Now, the homopolysaccharide is divided into two types based on their working principle. One is the structural homopolysaccharide and another one is the storage homopolysaccharide. Now, for the structural homopolysaccharide, this particular homopolysaccharide provide the mechanical stability to cells and organs of different types of organisms. As for example, we can take the chitin. The chitin helps to produce the hair and the nails production in the human, and also generates the hard outer shell, or we can say that the armor in various organisms for their protection. So okay, so now the next type of homopolysaccharide is the storage homopolysaccharide that helps to serve as the carbohydrate stores. That means it helps to store the carbohydrate. We can take an example that the glycogen, that means the glucose is stored in the liver in the form of glycogen. So this is the storage homopolysaccharide. Now the last one is heteropolysaccharide. So in this type, two or more different kinds of monomeric units bind with each other and forms this heteropolysaccharide. In the picture you can see that two or more colors of different monomeric units are present and binds with each other and forms the heteropolysaccharide, and this is also have two types, that is called the branched and unbranched based on their structures. Now we have also two special types of carbohydrates that are the reducing and non-reducing sugars. So how we can identify these two special types of carbohydrates? Now for reducing sugars, we can see that the carbohydrates which reduce the Fehling and Benedict's reagent. So now what are the Fehling's and Benedict's reagent? Fehling's reagent is a chemical reagent used to differentiate between water soluble carbohydrate and ketone functional groups, and it is also used as a test for reducing sugars and non-reducing sugars to identify. So okay, so this is the Fehling's reagent, and for the Benedict's reagent we can say that this is also a chemical mixture of some chemical reagents of sodium carbonate, sodium citrate and copper sulfate pentahydrate, and it is mainly used in place of Fehling's solution to detect the presence of reducing sugars. So okay, and we can also identify the reducing and non-reducing sugars based on their free aldehyde and keto group presence. So if we take an example of maltose and lactose for the reducing sugar, we can see that they have the acetal and as well the hemiacetal group in their structure, and in the hemiacetal point we can see a free CHO group, that means a free aldehyde group is present. So how the aldehyde group is present here? The hemiacetal means when an alcohol is combined with and is mixed with an aldehyde, then the hemiacetal point is produced. So we can say that the reducing sugars have the free aldehyde group in their structure or free keto group in their structure. Now, if we take a look at the non-reducing sugar, we can see that the non-reducing sugars have no free aldehyde or keto group in their structure, as we can see in the example of sucrose that there is no hemiacetal group. That means they lack the hemiacetal functional group. That means they have no free aldehyde or keto groups. So, thus we can identify the reducing and non-reducing sugars based on their structures of free aldehyde or keto groups, besides their response on the Fehling and Benedict's reagent.